Welcome friends, we will discuss about reproduction in bacteria. So please go through part 1 and part 2 of the video to have all the general idea about the bacteria. Now the last part is reproduction in bacteria, which is very important, how they reproduce. So they reproduce by vegetative method or by sexual method or asexual method. Okay, three methods of reproduction are there. In case of vegetative mode of reproduction, it includes budding, fragmentation and binary fission. Fragmentation and binary fission. So what is budding first of all? Let's start with budding first. So what is budding? Budding is the formation of the bud. Okay, it's a kind of small protuberance. So small protuberance which come out, and this protuberance is called as bud. Where it develops from one end of the cell. I'll show you the picture also, I'll draw it. Develops from one end of the cell. And once the bud develops, the genome replication follows. Okay, the genome replication follows in the new protuberance which develops. So genome replication will follow and a copy of the genome set will be formed. Okay, so copy of the genome gets into the bud. Then the bud will enlarge, then the bud enlarges and it becomes daughter cells. Gradually the bud will enlarge and become daughter cell. It will become daughter cell. And this daughter cell gets finally separated from the parent cell. Gets separated from parent cell. So this is about budding, how it looks like. So if this is a bacterial cell, okay, if this is a bacterial cell which has the genome structure inside and few organelles like ribosomes, and the nucleoid which is there, it will replicate. The nucleoid, the true nucleus is not present, so nucleoid which is there, it replicates. And there is a development of new nucleoid as well as a protuberance. So here is the development of new nucleoid which is going to develop and protuberance. Okay, then this will move into the protuberance. This new nucleoid will move into the protuberance. This is the old nucleoid and this is the new one which move into the protuberance. Then the young bud will separate. The young bud, it separates from the parent cell. Okay, so what we see in the second step, number one step that the nucleoid will replicate. Second step that the new nucleoid moves into the bud. New nucleoid moves into the bud. So this is a young bud. And the last is the formation of daughter cell. So this is the process of budding in bacteria. Okay, so next type of vegetative reproduction is fragmentation. What is fragmentation? Like the fragments which develop into the new bacterial cell. Okay, so this fragmentation mostly it happens during unfavorable condition. Okay, and the bacterial cytoplasm which is there inside the bacteria, the not cytoplasm, I'm sorry, the bacterial protoplasm, they undergoes compartmentalization. They form compartments. Okay, they undergo compartmentalization and fragmentation, and thus they form the fragments. And fragmentation results in the formation of minute bodies called gonidia. Okay, and these gonidia, when they come under favorable conditions, so under favorable condition, what happens? Under favorable conditions, these gonidium grows into new bacteria. Okay, these gonidia which is formed, they grows into new bacteria. Understood? And this becomes apparent that prior to fragmentation, the bacterial genome has to undergo repeated replication. Okay, so prior to fragmentation, the bacterial, because each fragment must get the genome. Okay, so prior to fragmentation, each bacterial cell, each bacterial genome, means the number of fragment is equals to the number of times the genome will be replicated, right? Has to undergo repeated replication so that it can transfer genome to each of the fragments, and each of the fragment can develop into new bacterial cell. Repeated replication so that each fragment gets a copy of it. So this is the process of fragmentation. The third is binary fission. What happens in binary fission? Now, binary fission is the most common type of vegetative reproduction, and this happens during favorable conditions. During favorable condition, binary fission occurs, in which the cell divides into two smaller daughter cells. Okay, a single cell divides into two daughter cells. Okay, and the single cell divides into two daughter cell, and during this process also the bacterial chromosomes get attached to the cell membrane and replicates to the bacterial chromosomes. Okay, so as the cell enlarges, the daughter chromosomes get separated. Okay, so if I draw and then I explain, it would be better for you to understand. So what happens? Say for example that this is a cell. This is a bacterial cell which has the nucleoid genetic material, and it has a cell wall. This is a cell wall. This is again inner to the cell wall is the cytoplasmic membrane. This is the cytoplasm. This is the cell wall. This is cytoplasmic membrane or plasma membrane, and this is the replicated DNA. Okay, I am drawing DNA with green color. So this is replicated DNA. Okay, and we can say that it is nucleoid, and this is replicated DNA. Okay, now in the first step, what happens? In the first step, there is cell will replicate its DNA. So nucleoid replicates, I can say that nucleoid replicates. In the second stage, in the second step, the cytoplasmic membrane elongates. The cytoplasmic membrane elongates and separates the DNA molecules. So DNA molecules are now far apart. Okay, and so I write here that the cytoplasmic membrane elongates, separating DNA molecule. This is the second step. What will be the third step? That there is the separation. Okay, I will draw here that I will draw for cells for 3, 4, 5 also, otherwise I have to change the color in the meantime. Okay, so number 4 is again separation of the cells, and number 5 is 2 daughter cells are formed. Don't go with the size of the cell right now. Just remember that the size of the cell is smaller than the parent cell. And it will be a double layered plasma membrane and cell membrane. I am not drawing that. Okay. So, in the second step, it is the cytoplasmic membrane elongates, it separates the DNA molecule. In the third step, a cross wall is formed. In the third step, a cross wall is formed. Okay, in the third step, cross wall is formed, and the membrane invaginates. Okay, here I also write numbers. It will be easier for you to track. The membrane invaginates. Invaginates means it's a kind of invagination, that it comes nearer to each other. Okay, now number 4 is cross wall, which started to form in step number 3. It completes. Cross wall formation completes. Completed. And in the 5th step, daughter cells are formed, which are now independent. Okay, so this is about binary fission, and this is about the vegetative reproduction in bacteria. Now coming to asexual reproduction. So what about asexual reproduction? So this asexual reproduction, it takes place by endospore formation. What happens in asexual reproduction is endospore formation takes place. Okay, what else? Conidia or zoospore formation takes place. This is asexual reproduction where spore formation takes place. So first of all, what is endospore? What is endospore formation? So endospore, what kind of spores are these? These are resting spores. Okay, these are resting spores. They are formed in some gram-positive bacteria. Which kind of bacteria forms this endospores? These are formed by gram-positive bacteria. Example, Bacillus, Staphylococcus, okay, Streptococcus, Clostridium, etc. When during unfavorable condition, in which condition are these endospores are formed? They are formed during unfavorable condition, when the conditions are not favorable for bacterial growth. And these endospores, where are they formed? They are formed within the cells. They are formed within the cells. And how it is formed? So during the process of formation of endospore, a part of the protoplast become concentrated. So I write here the process of endospore formation, which is a part of protoplast becomes concentrated around the chromosome. Around the chromosome, a part of protoplast they become concentrated around the chromosome. So that part will become the endospore. Okay, and around that a hard resistant wall is secreted. Okay, a hard resistant wall is secreted around it. Just imagine the structure. I'll also try to show you. So a hard resistant wall is formed around it, and the rest of the bacterial cell, the rest of the protoplast, the rest of the bacterial cell or the content, it will degenerate. So interesting to know, right? Degenerates. Only that part around which the hard core resistant wall is formed, that is living, and that is forming the endospore. So since the wall is very much resistant, which makes the endospore very resistant, and these endospore, they are very resistant to extreme physical conditions. Okay, so these endospores, what we talked about, that these endospores, they are very resistant under unfavorable conditions. Okay, they are very resistant under unfavorable conditions and under extreme physical condition, under extreme conditions like physical or chemicals. And during favorable condition, these spores will germinate. Okay, they will rupture during favorable conditions. During favorable condition, what will happen? The spore wall will degenerate. The spore wall, which is very resistant, the spore wall gets ruptured. The spore wall gets ruptured, and the protoplasmic mass give rise to new bacteria. And the protoplasmic mass which is there give rise to I'm sorry, give rise to new bacterium. So what we talked about endospore, that they are formed by what? They are formed by gram positive bacteria. Okay, gram positive bacteria, where are they formed? They are formed within the cells. What is the process? The process is they will form within the process is the protoplast becomes concentrated, okay, around the chromosome, and this will form the hard resistant wall and forms a hard resistant wall around it, and the rest of the part will degenerate. So rest of bacterial cell degenerate. Got it? What happens here in case of, and also these endospores are what? They are resting spores. Means they will only form the new bacteria in the favorable condition, otherwise not. So would you like to study endospore with the picture? Yes, sure. Okay. So when we have seen that this bacterial cell which develops by binary fission, so starting initial few steps will be similar. Okay, so what are these steps? If I write these steps, what is What are these steps? Number one is DNA is replicated. Whenever a replication or any kind of reproduction occurs, there is DNA which is replicated, like in case of sexual reproduction or in case of vegetative reproduction, first is DNA is replicated. Okay, then the DNA aligns along the cell's long axis, means the DNA will move to the two corners so that the wall will form in the between. So DNA is aligned along the length of the cells. I'll draw it along the length of the cell. So see here that DNA is replicated if this is a cell, and here the DNA is replicated. Got it? Then in the second step, this will align along the length of the cell, means one will be here and one will be here. Okay, now the third step is cytoplasmic membrane will invaginate. There is invagination. So cytoplasmic membrane, not the cell wall, but cytoplasmic membrane will invaginate to form 4 spore. This is 4 spore, which is before the formation of endospore, it is 4 spore formation. Okay, so I draw here again. This is the formation of invagination by the cytoplasmic membrane. Okay, and these are the nucleoid. Then the cytoplasmic membrane grows and engulfs the 4 spore. As I said that some part will form a coat and the other will degenerate. Okay, so here in the fourth step, what will happen is that there is a formation of hard coat around the chromosome. Okay, and the rest part will degenerate. So this is the first membrane which is formed. Okay, this is the first membrane. This is called as 4 spore because this is the one which is going to form the spore. So 4 spore. Okay, so this is the first membrane. Okay, and this is the second membrane. So this is the first membrane. I will draw it clearly. This is the first membrane and this is the second membrane which forms inside. Okay, and this part will degenerate as I discussed. Now the fourth step, I write here that the cytoplasmic membrane will grow. Then the cytoplasmic membrane grows and engulfs the 4 spore within. And engulfs, so this membrane is again the cytoplasmic membrane. Okay, and engulfs the 4 spore within. The 4 spore within. And forms a second membrane. Okay, and this one will degenerate. Now in the fifth step, a cortex of calcium and dipicolinic acid is deposited between the membrane. So a cortex of calcium and dipicolinic acid is deposited between the membrane. So if this is a structure and if this is which remains and this is the layer which forms, this is called as cortex. Okay, and calcium and dipicolinic acid and all deposits here. Next is spore coats forms around endospore. So now this becomes an independent unit where a coat develops. Okay, now you are seeing that here it is attached. Here it is attached with the cell wall, but here a spore coat is there. This is the spore coat, which is the endospore coat which is formed here. Okay, so spore coat is formed around endospore and this is called as spore coat. Then maturation of the endospore will take place and completion of the spore coat. So maturation, so spore coat is still not mature. It will mature. The endospore is not yet mature. Okay, so maturation of endospore and completion of spore coat. Completion of spore coat and increase in resistant to physical damage, which makes it resistant to damage. So how it will look like? It will be even thicker coat. Okay, it will be even thicker coat. So this is the nuclear structure. This is the even thicker structure which is now more resistant. Okay, this is now more resistant to any kind of physical damage. Hope it is making sense. So this is the endospore structure. Got it? Which is more resistant. Now the endospore will be released from the original cell. Released from original cell. So this will be released from the original cell. Okay, and this is the outer spore coat. This is now more resistant. Clear? So again another thing is conidia. So second kind of spore is conidia, which are formed in chains. They are formed in chains. Okay, they are formed in their kind of branched kind of structure which are formed in chains. Okay, where you can see these spores are formed in chains, in groups. Okay, and they each have the capability to form the new bacteria. Each have the capability to form the new bacteria. Each can form new bacteria. And in which kind of bacteria are these conidial spores are formed? So the example is Streptomyces. Streptomyces forms conidia, and they are found in filamentous bacteria like Actinomycetes. Example is Streptomyces. So they are found in filamentous bacteria. So this is a filamentous bacteria. Example Streptomyces. Like Actinobacteria. mycetes. Okay, so this was about the asexual mode of reproduction. Then we will move to sexual mode of reproduction. But before moving to sexual mode of reproduction, we will start with the mycoplasma. Because it's a very small topic, so we will cover it quickly. What are mycoplasma? So these mycoplasmas, they are a genus of bacteria. What are they? They are the genus of a bacteria, and the important characteristics of the mycoplasma are they lack cell wall. Okay, they lack cell wall around their cell membrane, and they are called as molecules. Molecules meaning what? Molecules meaning soft skin. Soft skin. So there was a scientist named Nocard and E.R. Rocks. Nocard, C-A-R-D, Nocard and E.R. Rocks. What they did, they studied pleural fluid, that is fluid from the lungs of the cattle suffering from bovine pleuropneumonia. They discovered very different organisms, okay, and they found to be different forms of some structures which are spherical, thin branching filaments, astroid, etc. Okay, and similar pleomorphic structure was isolated from other organisms. They named, so they have discovered some pleomorphic structures. And named them PPLO. What is PPLO? Which is pleuropneumonia-like organisms. Pleuro, again second P is pneumonia, like organisms. So while they were studying the cattle, they were studying the pleural fluid of cattle, and they discovered some kind of which cattle they were studying. So cattle which was suffering from pleural fluids they were studying. Which cattle? The cattle suffering from bovine pleuropneumonia. Suffering from bovine pleuropneumonia. Pleuropneumonia. And they discovered several organisms which have the pleomorphic structures. Okay, so what are the characteristics of mycoplasma? What are the characteristics of mycoplasma? The first thing is they are unicellular. Like bacteria, they are also unicellular. Difference is they do not have cell wall. Okay, so they are unicellular. They are also prokaryotics, which is similar as bacteria. They are the genus of bacteria which are slightly different from rest of the bacteria. Okay, so they are usually non-motile in nature. They are usually non-motile in nature. They form fried eggs-like colonies. They form fried eggs-like colonies. They are pleomorphic in shape, so they are highly pleomorphic kind of shapes, and they can be filterable through bacterial filters. We can filter it through bacterial filters. So they are filterable through bacterial filters. Cell wall is absent, which we know. They have lipoprotein triple layered plasma membrane. They have lipoprotein triple layered plasma membrane. So, and both DNA and RNA are present. Both DNA and RNA are present. Otherwise, how it will replicate, right? So, both DNA and RNA are present. They are resistant to antibiotics because cell wall is not there, so antibiotics will not act. So, resistant to antibiotics. What antibiotic? Example, penicillin. Another is they are inhibited by tetracyclines. How tetracycline? Because tetracycline is not acting on the cell wall. It is acting on, because tetracycline is acting on the metabolic pathways. Tetracycline affect metabolic pathways. That is why tetracycline can inhibit this mycoplasma, and then they are mostly free living. Parasites or saprophytes, and they are very small in size. They are around 30 nanometer to 0.2 micrometer. To 0.2 micrometer in diameter. What about the other characteristics? These mycoplasma cells, they divide unevenly into very minute bodies. Okay, so they divide unevenly into very minute bodies called elementary bodies. What you will call this minute bodies as? Elementary body or minimal reproductive unit The size of 330 nanometer to 450 nanometer, like for example, since the diameter is so small, size is also small, these mycoplasmas, they enlarge to form filaments. Okay, or mycelia, and chains of minute spheres. Like for example, you have seen in conidia that the chains of spheres are formed and each can give rise to bacteria. Same here in mycoplasma also. Okay, and how this small spherical shaped, how this small spherical shaped mycelia or chains of spheres are formed? They are formed by So these conidia, they are released by rupture of membrane. So the best studied genera are Mycoplasma, Ureaplasma, Mesoplasma, Spiroplasma, Entomoplasma, Aneroplasma, and Asteroplasma. So these are the best studied genera. So that's all for Mycoplasma. So we will continue with the sexual reproduction part, which is again very important for you to know, because the most of the important topics are from the sexual reproduction of bacteria.